This video is going to explain how cells communicate with each other and how materials are transported in and out of cells. So here we have a picture, a diagram illustrating a little section of a cell membrane, and let's just label some of the parts so you remember what cell membranes are made out of. Again, this first part is the phospholipid bilayer, and of course the round phosphate heads are hydrophilic, or like to associate with water, and the fatty acid tails are hydrophobic, or don't like to associate with water. And that's why they associate in this bilayer format where the fatty tails are facing each other, excluding water molecules, and the phosphate heads are facing the water on the outside of the cell and on the inside. So we can label that outside the extracellular environment and the inside the intracellular environment. Now I have shown also a cholesterol molecule that helps to deal with membrane fluidity. Alright, so imagine that you have these phospholipids making up the membrane. If it was only phospholipids, nothing could penetrate in or out of the cells. Maybe perhaps small fat-soluble or lipid-soluble molecules could make it through, but none of the larger molecules or no water-soluble molecules could make it into the cell, or any waste products out of the cell. So scattered around the cell, throughout the cell membrane, are proteins that allow things in and out. We call this understanding, or this theory, or this model of how cell membranes work, the fluid mosaic model. Because the membrane is like a mosaic, a bunch of small pieces, little proteins, little phospholipids all scattered around, but they're fluid. They can move side to side. They're not anchored in any one particular place. Okay, so imagine you have nutrients that the cell needs, maybe some sugar or some nutrient that the cell needs, and it's floating around outside the cells. Could be floating in water outside the cell, or perhaps in a bloodstream, within a blood vessel outside the cell. So if these nutrients are out there, and there just happens to be a protein with the right shape to allow these molecules through, then they can come inside. So for example, let's imagine these green molecules might be something like a sugar or an amino acid, some nutrient the cell needs. They can be brought inside through these proteins. We're going to call this kind of protein a transport protein, because its job is to transport materials in and out of the cell. So here's our little square sugar. Now, could another one come in? Sure. Let's just bring another one inside. Of course, we have to realize that that sugar is no longer outside, so we'll erase those. A couple of sugars brought out. Could bring a third one in? Sure. How many can be brought in? See, here's the problem. If we pack too many inside the cell, there's more of these molecules inside the cell, and they're crowded, they're bumping up against each other, and really the shape of this transport protein is just a channel, just a hole right through it. Sometimes these proteins are also called channels, and they could allow these molecules to escape back out. So that might happen. Maybe another one goes out. So if you think about it, how many are going to end up on the inside compared to the outside? If there's more outside, they're likely to move inside. If there's way more inside, they're more likely to move outside. So actually the molecules keep moving back and forth all the time. But eventually they'll kind of even out to where you have the same amount coming in as going out. In other words, they'll reach an equilibrium. Now this kind of process, it's called facilitated diffusion, because diffusion is just the movement of molecules from where they're crowded to where they're not crowded, or from where they're highly concentrated to less highly concentrated, down a concentration gradient. However, since these molecules can't just move, because this cell membrane is a barrier in the way, they have to go through a protein channel or a transport protein in order to get into the cell. So they call it facilitated diffusion, which means, well, facilitated means assisted or helped. So it's just assisting the diffusion because it can go through that little opening. However, if you think about this process, this doesn't make a lot of sense for something that the cell really needs, something like sugar that's going to provide it energy. It doesn't want to get by with only half the sugar available. If it has to leave half of it outside the cell. For something like glucose that the cell really needs to power itself, it's going to want to get all of it. So let's draw let's make some changes. Let's say this green square is actually maybe something like carbon dioxide. As carbon dioxide as the cell does metabolism, builds up carbon dioxide within the cell. As the carbon dioxide builds up, more and more in the cell is going to mean it's going to transport more and more outside so that it gets rid of it. The nice advantage of facilitated diffusion is it doesn't take any effort, any energy from the cell to do this. It just is an automatic process because molecules are constantly in motion. It'll just do it automatically. If we think of glucose that the cell needs, let's explain using this next protein in the membrane how it might get all of the glucose inside the cell. So here's some glucose molecules outside the cell, and if you notice this transport protein has a little shape that will fit the glucose. So imagine glucose binds into that opening. If the glucose molecule binds in, it could alter the shape of that protein somewhat. Now imagine that this molecule, this protein, has think of it as having a little hand that can reach out and grab those glucose molecules and throw them into the opening. But what's going to power that hand, change the shape of that protein to make it reach out and grab something? We're going to need a molecule that provides energy to change the shape of that protein. So let's, in this active site at the bottom of the protein, let's attach a molecule that provides energy. ATP, for example, could do that. It's a commonly used molecule that provides energy. So let's say there's a molecule of ATP. It binds to the protein. Protein reaches out, changes shape, grabs a glucose molecule, throws it into the protein, fits in the active site into that shape. That changes the shape of the protein further until it looks like this. And the glucose molecule falls out and is released into the cell. Then the protein can change back to its original shape again. And notice that the ATP molecule has also left the active site, and as the protein changes back to its original shape, now that it's missing the glucose, missing the ATP, it'll revert back to its original shape. And now it's ready for that part of the protein, the hand, to reach out, grab another glucose, power it up with more ATP, and it's ready to repeat the process all over again. This can happen as long as there's a supply of these ATP molecules supplying energy to the protein. And in fact, probably a good percentage, maybe even half of the energy that our bodies produce, that our cells use, is used in this process of pumping molecules into a cell or out of a cell against their concentration gradient. So actually, this can bring all of the glucose in. Even though there may be a lot already in the cell, it can continue to bring more in, which would be against what diffusion would like to do. So this process requires energy from the cell to do this, and it's called active transport. But it can result in the bringing in of many molecules into the cell where facilitated diffusion wouldn't be able to accomplish that. The next protein in line, notice it doesn't have an opening that goes all the way through the protein, so it's not a transport protein. Instead, the job of this kind of protein is to receive messages. It's going to receive signals from some other cells. So we're going to call this a receptor protein, or a receptor. And notice that it does have an active site, similar to an active site of an enzyme, where a certain shape of molecule can fit in, and only the certain correct shape would fit in and activate this receptor. So in our example, of course, which shape is going to fit into that receptor? You got it, the triangle. So if that triangle molecule fits into the receptor, it could alter the shape of that receptor, send up some tremblings, alter the bonds, and cause this purple molecule at the bottom to drop off. Now, that purple molecule, we could call that a second messenger. So what's actually happened is we've been able to send a message into the cell without actually crossing the cell membrane. We've just attached a signaling molecule, and then a second messenger detached from the receptor protein and carried the message inside the cell. The signaling molecule, we could call that a signal, or sometimes you'll hear the word ligand. That's the molecule that binds to the receptor. The second messenger molecule can drop off. In many cases, it might combine with another molecule in the cell. In this example, these two get together to become a new molecule, a combined molecule that looks like this. And of course it's going to have a function, so it could come over here, slice this molecule in half. Each of these two molecules can then go and combine with another protein, in this case perhaps an enzyme. These might be an example of a coenzyme that would enhance the function of these enzymes. And then these enzymes are free to move into the cell and do whatever function they're designed to do, perhaps even moving into the nucleus and activating some DNA. This process of second messenger activating another protein, activating another molecule, activating another one, that's called a signal cascade. And depending on which proteins are inside the cell, the signal or ligand can cause lots of different reactions depending on the type of signaling cascade proteins that are available inside different cells. The last kind of protein is called a marker, and its job is simply to advertise what kind of cell this is. Often they come with carbohydrates attached or lipids, so this could be a lipoprotein or a glycoprotein. Think of this as a little identity badge. Okay, so it's a marker protein. And in a way, these are responsible for our immune system being able to recognize which cells are us and which cells are not us. Every cell in a body contains these marker proteins. They advertise that this is us, so that, say, a bacteria or a virus, something without those specific proteins gets in, our immune system cells recognize that as an invader and they attack. It also is important to understand for as far as organ transplants, that these marker proteins create issues of organ rejection. Say, for example, somebody needs a kidney transplant. You can't just give anybody's kidney to them because they would have the wrong marker protein. They need to try to match up as many of the specific markers on cell types as they can. For example, everybody's heard of blood types. Blood cells may only have a couple of different marker proteins that would be recognized by immune systems. But, say, a kidney cell might have a dozen, and they need to match up as many of the dozen as possible to make sure they're the same. Which is one reason why if you can't get them all to match up, you may have to try a transplant and then give someone immunosuppressant drugs that just try to keep their immune system from functioning and rejecting that organ. Otherwise, they think of it as, like, you can imagine they would respond to it like a giant bacteria, and they're all, attack, it's not us. So that's one reason why these marker proteins are important. And these are the main categories of types of proteins that are on our cell surfaces. They allow things to get in and out, they allow messages to be passed from cell to cell, and they allow cells to recognize which kind of cell it is and where it should be in the body and how they should respond to it. Hopefully you learned a lot about how cell communication and cell signaling works.